What happens if I drip ordinary kiwi juice directly onto thousands of live bacteria from stagnant water? Let's take a look at their reaction under the microscope. Right now, under this lens, there could be over 1,000 tiny organisms swimming around, spinning, hunting, just living their tiny lives. And in a few seconds, their whole world is about to change. Let's watch what happens. Okay, here's our water sample. I collected this from a stagnant pond a few days ago. Stagnant water might look gross to us, but under a microscope, it's basically a tiny universe. Ponds like this are usually packed with all kinds of microorganisms. Some swim fast, some barely move. Some look like they're spinning in circles for no reason at all. I can't tell you exactly what species we're looking at. Without lab testing, we really can't be 100% sure. But look at that movement. That is life. Real, active, moving life. Now let's bring in the kiwi. Kiwi juice is naturally acidic.
It also contains a special enzyme called actinidin. You've probably tasted its effect before, even if you didn't know it. It's the same enzyme that makes kiwi great at tenderizing meat. So what happens when that acidic, enzyme-loaded juice meets a slide full of living microorganisms? Let's find out. Here comes the drop, and there it is. The juice is spreading across the slide. Watch the edge of that liquid right now. Watch closely. Wait, did you see that? Something near the edge just froze mid-swim. Let's zoom in. Okay, this is fascinating. A few seconds ago, this whole area was full of movement. Now look. Some organisms have slowed down. Some are barely twitching. But over here, over on this side, we've still got a few holding on, swimming like nothing happened. Why would that be? Here's the thing about a microscope slide. It's not one uniform space. The kiwi juice doesn't hit every organism with the exact same force at the exact same time. Some organisms are closer to where the drop landed. Some are farther away, in water that hasn't fully mixed with the juice yet. So we're basically watching a wave of change roll across this tiny world in real time. Let's keep watching. Right here, this one is still moving pretty fast, almost like it hasn't noticed anything's wrong yet. But over here, completely still. That's the kind of contrast that keeps this footage so oddly hypnotic. Some organisms clinging to motion, others just stopping. Now let's slow things down and actually talk about why this might be happening. And I want to be honest with you here, because I'm not going to pretend we know for certain. Without lab equipment, we can't run the exact tests that would give us a definitive answer. But there are a few likely explanations based on what we know about kiwi juice. First, acidity. Kiwi juice is fairly acidic. Many microorganisms are extremely sensitive to pH. Even a small shift toward more acidic conditions can disrupt how their cells function, and that can affect their ability to move. Second, the enzyme actinidin. This enzyme is known for breaking down proteins. Since a lot of the machinery inside a living cell is built from proteins, an enzyme like this could interfere with the structures an organism needs in order to move or survive. Third, osmotic stress. This is just a sciencey way of describing what happens when the balance of water and dissolved substances changes suddenly around a cell. Cells are very particular about that balance. When it shifts fast, it can put the cell under serious stress. And fourth, simple environmental change. Sometimes it's not one single dramatic cause, it's everything at once. New chemicals, new acidity, new pressure on the cell, all happening in the same few seconds. That combination alone could be enough to slow things down. So which one of these is the real reason?
Honestly, we can't say for sure just from watching under a microscope. That would require proper lab analysis. But watching this reaction happen live, it's still incredible. Let's check back in on our slide. Whoa, look at this area now. A few minutes ago, this whole region was full of tiny swimmers weaving in and out of view. Now it's almost silent. A handful of organisms are still twitching, still trying. Wait, did you catch that one in the corner? It just kicked back to life for a second. That's actually one of the wildest parts of doing microscope experiments like this. It's rarely instant. It's rarely all or nothing. You get these little moments of resistance, tiny bursts of movement from organisms that are clearly under stress, but haven't fully shut down yet. It's honestly kind of beautiful, watching something so small put up a fight we can barely see with the naked eye. So here's where we are. Most of the movement across this slide has stopped. A few organisms near the edges, in areas where the kiwi juice hasn't fully spread, are still going. And based on everything we just talked about, the acidity, the enzyme, the osmotic stress, the sudden environmental shift, any one of those, or some combination of all of them, is likely responsible for what we're seeing. But here's the honest truth. This is a home experiment, not a lab study. We're observing, not diagnosing. If you want to know exactly which factor caused exactly what, that takes real laboratory testing, controlled conditions, and proper equipment. What we do have, though, is footage of something genuinely amazing. A tiny, invisible world reacting in real time to something as simple as a piece of fruit from your kitchen. I think that's what I love most about this kind of experiment. You don't need some crazy expensive lab to witness something incredible. You just need a microscope, some curiosity, and a willingness to ask a weird question like, what would happen if I did this? If you enjoyed watching this tiny world react in real time, make sure you subscribe. This is just the beginning. I've got a lot more microscope experiments coming, and honestly, some of them are even stranger than this one. So stick around. The next drop might surprise you even more. I'll see you in the next video.